C, D, E, F, G, H. There's Ryan, he's H. Okay. You're all in your groups. Put your clothes away. I mean your books, not your clothes. Okay, put all your stuff away. Okay, you guys ready? Hold on. Hold on. Let's just keep it. Max. Under control. Thank you. Calm as a river. Okay? Okay, you ready? Let's go. Where's the representative of group number 1? I mean A. The thing is that if you pull, can I have your attention, please? And let's cut it out. Eden, that's it. Marvin, that's it. Marvin, silence. Okay, if you pull, you're going to pull 2 little tags. If you pull the same one, obviously you got to put one back in because you can't challenge an answer on the same thing. So let's have an answer question for group number 1. And can you, Daisy, and please write these down, Aicha, because I'll never remember. Pull it out. Question number 3 for the answer. Keep it with you. Question 3 for Group A. And question 4 is the challenge for Group A. Let's go to B. Group B. There you go. Rose. Group B has question number 7 as the answer question. And Group B has question number 2 as the challenge question. Great. Where's number C? Taylor. Dig deep. Answer number 4. Challenge number 6. Let's have Group E. D. Getting to be like Ingrid here. Hold it. Hold it, Michelle. Lauren, thank you. Question 2 for D. Is that correct? Challenge is number 5 for D. Let's go to E. Question answer number 1. Wow. And challenge number 7. And let's go to F. Don't look. Don't look. Answer number 5. Challenge number. Let's go to group number G.
We get what's left, right?
Yeah, what is it?
I don't know. You read them off?
Just a sec.
You guys keeping track?
Nope.
6 and 3. Okay, just a sec. I got a little record here. Group G has answer question number 6 and challenge question number 3. Okay. Group number A. What is your answer question? A, what is your answer question? 3. What is your challenge question? Group number B, what is your answer question? Challenge. C, and 4, challenge. Group number D, answer question. Challenge question. Great. E, answer question. 1, challenge question. Group number F, 5, challenge. Group number G, 6.
6 and 3.
Great. Okay, you guys now have 10 minutes to organize. Everyone ready? Where's group number A? A ready? Where's group number G? Group number G ready?
Okay? Yes. Yes.
Great. How about F? You guys ready? How about D? You ready? E? B? C? A? Okay. Great. I know all of you are going to do really well. Now remember, nice little bullets passing from one person to the next, just like you do on your quizzes and everything else that you've done so great on. Now keep in mind one thing as we discussed with the midterm oral. Pay attention, because you never know when you're going to be called on, even though you're not in the group. Okay? And secondly, again, just like the midterm oral, those of you that go last, please don't do any work on your questions because it's not fair to the people that go first. And so that's really sort of the little mini rule. And about halfway through, we'll take a biology break. And so we're not just sitting there and holding it for the whole time, which I don't do very well at. In any case, let's try question number, let me see what we should do first. How about question number 1? Fail-safe quiz question number 5. The answer group is E. The challenge group is 7. This deals with forensics, DNA testing, and all of the above. So let's do that particular question, and the DNA profile is on the screen if you need it, and there's pointers there, and please use the blackboard to write down your answers, okay? So use the blackboard right over there, yeah. The whiteboard. Just use that whiteboard there, okay? You now have exactly, let me shut this off, 9 minutes. Go.
All right, so question 1, DNA fingerprinting using crime scene materials
Slow
and
down.
suspect cheek swabs. All right, so we have to collect samples of DNA from the crime scene, either blood, skin, or hair, and we have to collect cheek swabs from every suspect, and we have to collect a DNA sample from the victim. That's just to cover all the bases. And then we use 10 VNTR loci, and we use PCR. We have every primer for every VNTR loci. We use PCR to amplify the DNA, so we could have a bigger sample, so we can perform gel electrophoresis on it. Every well represents a DNA sample or a suspect, and after we do PCR to get all the different lengths, we do gel electrophoresis, and then we can compare the different lanes.
All right, and so what we do then is we compare the VNTRs of the suspects, the 3 suspects, to the VNTRs of the 3 samples. And if all 10 VNTRs, if not all 10 VNTRs match between a suspect and any of the 3 samples, that suspect could not have possibly committed the crime, or at least given the evidence that we have, and we must exclude them.
Okay. Because we do not have the allelic frequencies or the frequencies of the VNTRs in the actual population, we cannot convince a jury beyond a reasonable doubt that a specific suspect committed the crime. If all of the VNTRs were very common in a population, just by random chance, we could have one of the suspects match up with the evidence that was found at the crime scene. If all of the VNTRs, the percents in just the population were low, then we might be able to convince them beyond a reasonable doubt.
So after all our necessary tests that we performed, if you just take a look at the results, we can determine the genotypes of the different individuals, and I will be writing them down. So the suspect's genotype would be A2, A3, B1, B2. The victims would be A1, A3, B3, B3 because there's a bold line over there, which indicates that that specific allele is repeated twice, is homozygous. And for the semen, if we take a look at it, it's A2, A3, B1, B2. And the defense attorneys is A1, A1, B3, B3.
So the suspect is A2, A3, B1, B2. The frequencies of those, can you hold this for me so I can see the frequencies? The frequencies, A2 is 0.6, A3 is 0.3, B1 is 0.3, or sorry, 0.7. B2 is 0.3. So we have the equation that you use to calculate the frequencies for heterozygotes is going to be 2pq with with Q being the first allele and P being the frequency of the first allele and Q being the frequency of the second allele. So to apply, we're going to apply that to both of these. So it's going to be 2 times 0.6 times 0.3 times 2 times 0.7 times 0.3, and I don't have a calculator, but I believe it's about 0.15. And as a member of the jury, we could not convict the suspect or based on this information, because 2 alleles is not enough at all. 2 VNTRs is not enough at all, not enough information to reach a conclusion. And 0.15 is about 1 in 7. So 1 in 7 people from a random population would have this same frequency of allele.
Is that it?
That's it.
Challenge. Where's
Okay.
the challenge group?
So do you think that every arrested suspect should have to give up their DNA? Like, even if they're not convicted yet, do you think, like, it should be government mandated that everyone has to give up their DNA for, like, a possible thing here?
Do you mean everyone who's arrested or everyone in the population?
Everyone is arrested.
Okay.
I personally think yes, that they should. I mean, if you're suspected of committing a crime and you're arrested, it means that there's a reason for that. But I also think that if you're acquitted, that you should be able to remove your VNTR data from the database. So, yeah, that's my opinion.
Challenge?
Another question, you mean?
From all of you, yeah.
Okay.
Questions from everyone in your group.
What's a possible contamination, or what's a possible problem that could occur when you're doing this process?
The DNA.
Problem with this is band shifting. If when you perform a gel electrophoresis, the bands that would show a specific VNTR loci can shift and show an inaccuracy, show that it's not a match when it is.
And also a DNA sample can be contaminated and then amplified and it'll mess up the results of the test.
Challenge?
Also, what allelic frequency would you want to feel comfortable to acquit someone? Or convict someone?
Well, I mean, there's no specific like percent, but probably something like much less than 1%. I mean, just the reason we wouldn't want to convict this person is because 1 in every 7 people just randomly off the street that you pick could just happen to have this genotypic frequency. So we can't just convict this person. We don't have enough evidence.
Challenge?
What is the normal number of loci that you would use to look at in number of VNTR loci?
The normal number is 15.
15? Okay.
Correct.
And my other question was, if you have the DNA and you see that they're matching between a suspect and who we think is the one who committed the crime, and if you have a high enough frequency that you believe that this allelic frequency is very frequent out in the environment, out in the real world, does that mean you completely acquit the person, or do you keep them in for further testing?
Is that opinionated or?
No, I'm asking, like, what was your opinion in this case?
So you're asking, like, would we do more DNA testing?
Yeah. Yeah.
Like if we would take more DNA and perform it on more VNTR loci?
Well, going back to your your
Right.
answer.
Samantha, give a very specific question, please.
Is mine not? Okay. I was just
It's
asking.
ambiguous. Make it more specific.
What's another tool you could use for identification?
Be a little bit more specific.
For matching up allele frequencies.
You can look at RIFLIPs as well.
What did you say?
RIFLIPs?
Okay. Challenge?
Or besides gel electrophoresis?
Sequencing?
Is DNA testing enough, or would you also look at other forms of like kind of crime scene investigation, I guess?
Well, the more info, the better.
Also, what procedures would you do in at the crime scene to ensure no contamination?
There's specific procedures set out by, say, the FBI and other government organizations. Other organizations, just so there's as little contamination as possible. People wear gloves, people don't sneeze, people don't cough. They use sterile equipment just as much.
Any other challenge? Challenge from anybody in this room for this group? Anybody in the class? God, you're being... Oh, Tom.
Okay. You use that 2PQ. What equation is that from, and what assumptions might not actually hold true in the real world for that equation?
Reading my mind.
Excellent. That's an excellent question, Tom. This is the Hardy-Weinberg equilibrium principle. That's how we calculate. This would be for a heterozygote. This would be for a homozygote, dominant, homozygote recessive. And we would have to assume that there's random mating in the population, which is very unlikely.
From a biological point of view, what does the P mean and the Q mean relative to biology?
The two alleles, the two different alleles?
Yes, but when you come together in a Punnett square and you get this P squared plus 2PQ plus Q squared, which we've gone over, what does it mean biologically?
The parents, yeah.
The parents or?
The parent.
The gametes.
Thank you. The gametes of the parents are uniting. Correct. Anyone in this class have a question for this group? We have another 2 minutes. Tell me what amendment of the Constitution pertains to the question that Albert asked in terms of all arrestees giving their DNA samples. Is that permissible under the federal Constitution? And what would the police technically have to do in order to do that?
The Fourth Amendment, search and seizure, basically law enforcement cannot search for evidence without probable cause.
And in California, though, does one need a search warrant?
To get your take your DNA, or
yeah, to take your DNA.
it's in the Constitution, yes, the federal Constitution.
But in California,
No.
you could challenge it, but you don't need a search warrant. Okay, good. Tell me, Julie, with the polymerase chain reaction with that gel on the screen, how would you ever get band shifting? And what does band shifting really deal with mostly?
It deals with ineffective procedures, and you probably wouldn't with this because we have.
When
enough
would you get the
technology.
band shifting?
When people don't do it right.
Well, I don't know what it is.
When the gel has.
With PCR? Or,
No.
Brian?
No, gel electrophoresis.
Of what?
Of when after you've done PCR, when you're running.
You sure you
Well-
want to say you get band shifting with PCR?
Well, no, not with PCR. When you're performing gel electrophoresis, sometimes like one lane is more porous than another.
With what DNA sample? What kind of DNA samples from crime scenes? Ones that have been amped? Think about your article that
Okay,
you
yeah,
read.
ones that have been amplified.
Ones have or have not?
Have not been
That's
amplified.
correct. Because it's contaminated with all kinds of goop and stuff like that. That's absolutely correct. Okay, I have one final question, and then we're going to move on. How many alleles in an individual? You have to spread the work around here. I'm hearing from the same people. How many alleles in an individual?
There are 2 alleles per gene in an individual
Okay, why is that?
because one is inherited from the mother and one from the father.
Good. Tell me a little bit about the chromosome that came from the mother and chromosome that came from the father. How many DNA molecules are in those chromosomes?
The Y has 10,000,
How many DNA molecules are in those chromosomes?
20,000.
20,000
Well,
DNA
okay,
molecules.
There's 10,000 base pairs, right?
How many molecules?
Oh, there's one molecule.
How many molecules in those chromosomes after DNA replication, Marvin? How many chromatids after DNA replication?
1 chromatid.
After DNA replication.
Oh, 2.
How many DNA molecules in each chromatid?
2 or 1? No. 1.
Eric.
DNA.
Where are their name signs? Go ahead.
Oh, I'd say 1.
How many?
1
You sure?
DNA molecule.
Yeah. Final question for this group. Tell me about the structure of DNA, please. Major features. You better spread it out.
It's a double helix.
Okay.
Made of complementary base pairs. There's 2 purines and 2 pyramides. Adenine, thymine, cytosine, and guanine.
What else?
Sugar phosphate backbone.
What else?
Phosphodiester bonds between the sugar phosphate.
Great job. Thank you. Okay, terrific job. Really was. Come a long way since that oral exam. Let me see where we should go from now. Let's go to the back. Question number 2. Question number 2, the answer group is group D. Hold it. And the challenge group is group B. And this one is a take-home test, fail-safe question. And there it is. You have 10 minutes at the most, 9 minutes. You ready? Time is going.
All right, so in this question we are...
Talk to the class, not to me.
Hi, class. So in this question, in this question we get to, we're proving that DNA is the it codes genetic information for every living organism. So we have 6 different strains, 3 of them virulent, 3 of them non-virulent. What we're going to do is we're going to isolate and purify the DNA from the V1 strain. We are then going to introduce it to the A-virulent strain 2 and 3, just as it is purified. Then we're going to take another sample of the isolated and purified DNA from the V1 bacteria and treat it with DNase, which denatures the DNA. The first group is our positive control group, showing that the introduction of DNA is going to change the genotype and the phenotype of the transformed A2 and A3
Can you
strain.
try and write down 1, 2, 3, 4 on the whiteboard what your experiment is?
Okay.
And why don't you talk and someone can write? That's
Okay.
much easier.
So, 1, isolate DNA from V1 strain. 2, treat A2 and A3 strains with DNA from V1 strain.
A2 and A3.
You can mark that as your positive control. And then just positive control. Then treat DNA from the V1 strain, a separate amount of DNA from the V1 strain with DNase to denature the DNA. Sure.
I think you mean degrade the DNA.
Speak up.
Degrade the DNA, because denaturing would separate the two strands. Degrading is just like making the DNA ineffective. So degrade the DNA.
Good call. Treat separate colonies of A2 and A3.
Are you the only one answering, or you have other people in the group that's answering?
There are other people, but they're doing different parts of the question.
Okay, but this is the biggest part.
Would you like them to answer? Because I know
Whatever
they
you
can.
guys want to do.
Anyone else?
Exactly.
So after we treat separate colonies with the V1 strain, the results are that for the A2 and A3 strains that were treated with DNA that was not treated with DNase will produce the virulent proteins and become virulent cells. They'll become V1 cells, and the A2 and A3 strains that were treated with DNase they won't transform into the virulent strain. And this shows that DNA is the genetic material because the DNA was degraded and put when A2 and A3 strains were transformed with the degraded DNA, they did not transform. But when the A2 and A3 strains were transformed with the not degraded DNA, did transform into the virulent strains.
And just a quick note, you can't use the V1 DNA on an A1 strain because it naturally mutates to a V1 strain, and that wouldn't show any sort of definitive mutation. And part B?
Part B says, outline the specific observations expected in your experiment that will show unambiguously that DNA is the genetic material of the paralysis bacteria. So the first observation you would see is a change in the phenotype. Once the A2 and A3 strains are transformed, they will begin to produce the virulent proteins, and they will change into round, right? They'll change into round on the morphology. And also another trait is that is the stability of the DNA, and the Oh yeah. Okay. So it's the stability of the DNA because once the DNA the DNA that was transformed will pass on from offspring to offspring and will be seen in further generations. And And that also shows that the genes are, or the DNA is replicable from generation to generation.
Okay, so the properties of DNA are that it's universal because it's found in different types of bacteria and all species and humans as well. And then we have that it's phenotypical because once there's a change in the DNA, we saw, for example, in this case, that the bacteria changed the morphology from round to square when there was a change in the DNA.
Two other properties of DNA is that it's stable, that it passes on to the children, which is also the result of it being self-replicating. And also it's stable so that once that DNA is inserted into the formerly avirulent bacteria, it won't suddenly, once it's transformed into the virulent form, it won't suddenly just go back to its square formation.
Okay, so the molecular events that occur when A1 strain is transformed into a V3 strain with pure V3 DNA is that the V3 DNA will insert itself into the chromosome of the non-virulent bacteria, and then when replication occurs and the cell DNA is transcribed, when transcription occurs, you'll have the protein produced, the virulent protein.
When the DNA, when there's a change in the DNA sequence, when it's transcribed, there will be a different amino acid sequence, which in turn will produce a different protein. A central dogma will essentially, like, the DNA will result in a different protein.
DNA to RNA to amino acids and proteins.
Yeah. So to clarify, transcription goes from DNA to mRNA. Translation translates from that mRNA template into an amino acid sequence, which composes a protein. And that different protein causes virulence.
Is that it?
Yes, sir.
Challenge. Let's have a challenge from, let's see, group number B.
What was the first experiment that really dealt with this issue of DNA transformation? And could you explain it?
The first experiment that proved that DNA was the transforming principle was the Avery et al. Experiment, where there was smooth and round bacteria. The smooth bacteria was the virulent form. The rough bacteria was the the rough bacteria was the avirulent form. The smooth bacteria was the virulent form. First, he got the bacteria from infected mice, and then he killed. He got the virulent bacteria from the mice. Then he killed them, and then he mixed it together with the avirulent form in a test tube to prove that the rats weren't the ones who provided the transforming principle. And do you guys want to add, or do you want me to finish? You want me? Okay, so once he proved that it could occur in the test tubes, then he moved on using different things to destroy either the RNA, the lipids, and stuff to make sure that he separated the genetic material into different parts like RNA, DNA, and he destroyed them one by one and injected them into the mice to see which ones are infected. And transformation from the avirulent form to the virulent form occurred in all of them except for the ones that were destroyed by DNAs. Huh? Treated with DNAs.
Was there an experiment before that gave inspiration?
Be specific.
Which experiment motivated Avery et al. To do their experiment? What are the ideas for that? Which experiment led to those ideas?
Answer?
I think they first they injected it into the mice without separating each part of a cell, like the RNA and the DNA and the proteins alone, and that's what first encouraged them to do this.
It's your challenges here.
Can I just ask an unrelated challenge? Okay. How does the sequence and order of the nucleotide base pairs lead to a specific order of amino acid sequence, and what is special about it? And how does that kind of lead to how does that affect DNA as a hereditary material?
Because each sequence for every 3 nucleotide bases, it codes for a codon that codes for an amino acid, and the sequence gives the whole strand of amino acids, and it's degenerate, and there's 20.
What are the implications of having the codons being degenerate?
That different amino acids can be coded for, no, different nucleotides can code for the same amino acid because there's more than one sequence.
How does that affect like DNA replication and phenotypic expression?
It shows that not all mutations are going to directly affect the phenotype of the individual. It might just be a genotypic mutation.
On that same sort of note, when you're talking about the what makes DNA so specific and makes it the genetic material, if stability is one of the main, categories for that, one of the main reasons for that, how exactly do you go from the V strain to the A strain? Like, why does that happen? If if the genetic material must be stable.
So you're asking how DNA can transform if it's stable?
Basically, I'm asking about mutations and what kind of mutations.
Well, DNA is stable, but it's not perfect. Through mutations can occur during replication.
You have
and
2 minutes.
Causes Chris to answer the question.
If mutations were impossible, nothing would have evolved. I mean, we'd still be like nucleotide strands in like some pond or whatever, I don't know, but mutation drives evolution.
Specifically, what kind of mutations can occur, and how would each kind of mutation affect gene expression?
Single nucleotide polymorphisms, which I think include like deletions, insertions, single nucleotide polymorphism is a specific category, and then you have insertions and deletions, which are like one nucleotide being deleted or one nucleotide being inserted. And single nucleotide polymorphism is a change in the genetic sequence. It's like inserting a different nucleotide, and then you can have like whole sections of chromosomes that like mutate, or not mutate, frame shift.
Let's go. Could you guys split so I can see your board there? Would you be able to describe this experiment and tell the group in this room why this experiment will never work in 10,000 years?
Are you
so you go step by step by step and tell me why this experiment cannot work and cannot show that DNA is the genetic material. Your experiment.
Okay, are you referring to the fact that we didn't?
So you have isolated the DNA from
we've
strain.
isolated from
strain number 6.
Yeah, that we could have
So
treated
go ahead.
the cells.
Now wait. Then the second thing is treat A1 and A3 with what? You say, I'm just reading what you wrote. Treat A1
V1.
and A3 with V1. So you're treating it with V1 cells. You're mixing
No.
A3 and...
No, we're not, because it says isolate the DNA. We're only taking the DNA. Where you
So can you explain what you're... seriously, just simply. What are you doing in step 2?
We are treating A2 and A3 with isolated DNA from the V1 strain.
Isolated DNA.
Yes.
Why are you treating both of those strains?
I think we're supposed to I think it's supposed to be only A2. I remember, Michael, but I'm not sure.
Okay, that's okay. So it's with DNA.
Yes.
Is that correct?
Yes.
Why do you say that's a control?
It's a control because naturally it's seen that the V1 strain would convert to A1, so we wanted to show that it can go from V1 to...
What's it and what's that? And, I mean, can you be very specific about this?
Okay. okay, up here it says that.
We only have about 3 minutes, so Maura, you're going to have to be very specific on your answer.
The A1 transforms to, no, it doesn't. The V1 transforms to A1 naturally, as seen.
The V1 transforms to A1. You're positive about that? V1 is transformed to A1?
Yeah.
It's a transformation, or is it a mutation?
No, it's a mutation. I mean, like.
Okay. So can you please go over your experiment for me?
Somebody else want to do this, or am I going to?
So you have isolated DNA.
Okay, so you isolate the DNA from the
Wait, I got to see your board here. Step 2. You have
you treat the
A1 and treat the A1 strain with DNA from
the V1.
Okay.
No, we're A2 strain of bacteria.
We're in A2 now. Okay. And what is the result that you expect?
We expect that the A2 will transform into a V2 strain.
Into a V2 with V1 DNA.
Oh, V1, sorry. V1 strain.
Okay. Then you have DNA from V1. What is the next thing? I can't read it. Treated with what?
DNA.
With DNAs.
And then,
Result in
and then what do you expect?
We expect that that will not be transformed. The bacteria will not be transformed.
Is that a positive or a negative result?
It's a negative result because nothing happened.
So where's the positive control?
No.
And what if someone just spit into DNA and did something to it? How would that say that DNA is the genetic material? What about all the other molecules in the cell?
We could we could also isolate the RNA and.
Where is that in your experiment?
Okay, well, we were basing it off of the midterm where it does not indicate that, but you can also.
The fact that DNA
I don't know anything about a midterm. I'm just looking at an experiment.
Okay.
The fact that DNA is the only macromolecule introduced shows that it is making the difference because it's been isolated and purified.
Couldn't there be problems with that experiment? I mean, I can think about 20 of them since it's a negative result.
Okay, so you could isolate, like Mara said, the RNA and proteins and try using that to transform the bacteria, and then you would see a negative result.
What
because
did
nothing
Avery et
happens.
al. Actually do?
They separated macromolecules from the V1 or from the V1 strain. They separated it into lipids, proteins, DNA, and and then there was one more.
RNA.
And RNA, and they basically did this experiment, this process with all the macromolecules to show that the other macromolecules were not the ones that were transforming the V1s or the A2 cells.
Okay. Are there any challenge questions from anyone in the room or at Davis? Be very explicit. Eden, do you have one?
In step 2, what do you mean by treat A1 with V1 DNA?
Bacteria spontaneously takes up DNA from its environment and incorporates it into its own genome. So by treating, we're introducing the DNA to its environment.
Good. Any other questions from anyone in the room? Wonderful. Thank you very, very, very much. Good job. Okay. Let's go to question number 3. So that would be group A is the answer question, and the challenge question would be group G in Davis.
Yep.
Question number 3. The clock is ticking. 9 minutes.
Okay, so we want to construct a flesh-eating Staphylococcus genome library. So first we're going to isolate the DNA from the Staphylococcus cells, and we're going to digest it with E. coli 1 restriction enzyme. And then we're also going to digest our plasmid with the same E. coli 1 restriction enzyme. And for the plasmid, the enzyme is going to cut on the LAC-Z gene. So that's where we're going to insert our pieces of Staphylococcus cell DNA. So we're going to anneal it with ligase. And then we're going to introduce our recombinant plasmids into E. coli HB101 cells. And in order to find the ones that contain the Staphylococcus DNA, we're going to put the cells on plates treated with ampicillin and also Exgal. And ampicillin will kill any of the cells that didn't take up any plasmid. And the Exgal is going to be cleaved by any functioning LAC-Z gene. So if the LAC-Z gene is working, then the cell will turn blue. And that is not the one we want, because that means it doesn't have the flesh-eating, part of the flesh-eating genome. So we're going to take all of our white colonies that we have and mix them together, and that'll give us our library.
Okay, so for part B, they're asking you to find how many clones you need to make a genome library. So to do this, you have to find what's the probability of a restriction.
You write it out on the board.
Yeah, of course. So to answer this question, you have to find the probability that this restriction site will occur in this 1 million base sequence. So they give you that 80% of the gene is G's and C's. So you have 40% G's and 40% C's because they're complementary and they come in pairs. So that you would use the same process for finding the percentage of A's and T's. So you would have 10% A's and 10% T's. And so the probability of you getting a G is 0.4, and you multiply that by the probability of getting an A, which is 0.1, times 0.1, times 0.1, times 0.1, times 0.4, and you will get 0.0000016. No, too many zeros. I think it's like that. So that is, and then you multiply that by 1 million bases because you have 1 million bases in your gene. So you get 16 restriction sites. And so if you have 16 restriction sites, it means you have 17 fragments. So that means you need 17 vectors. And so part C, you want to identify the clone that contains your flesh-eating enzyme gene. So you want to reverse translate your enzyme because it's already pure. And then you use that sequence to create a cDNA probe. And so you label that probe, and then you rescreen your whole genome library. And oh wait, before you do that, you have to blot the genome library, and then you have to denature. Then you screen it with the probe. And then you expose it, or you identify which clone hybridized using autoradiography, and then you isolate that clone, you sequence it, and then you compare it to the sequence of your cDNA probe. And to double check, you inject this gene into a mouse to see if it dies within 24 hours.